So let's explore the structure of tRNA in this particular video. So we shall start by looking at tRNA. So what is tRNA? tRNA is the RNA that's involved in the translation, which recruits amino acids towards the ribosomes, you get, where we have the formation or decoding of mRNA. So tRNA, specifically, I want to say that ideally there are supposed to be 20 tRNAs, each specific for a given amino acid. However, in some organisms, we shall find that they have fewer tRNAs due to one having ability to carry more than one. So we're looking at the structure. We have a tRNA. Of course, it's formed as a primary structure, meaning it's formed as a linear structure, which has its 5' to 3' sequence. And later on, the primary structure can go on to form a clover leaf, which is known as its secondary structure. So secondary structure is a clover leaf, whereas it can also form a tertiary structure, which we're going to look at briefly. Tertiary structure is L-shaped. So its primary structure, like any other protein sequence, it's a single, it's a straight chain. Secondary structure forms this clover leaf, what we are looking at. So this is its secondary structure, what we are looking at today. Then can also go on to interact to form an L-shaped tertiary structure. So looking at the structure here, it has these what you call the loops. These bulgings you see here, those are called loops. So if we are to count, we have 1, 2, 3, 4 loops, a total of 4 loops. So why are loops formed? The reason as to why we form loops is we are going to see it shortly. Then we have these regions here where we see as if they are parallel sequences. These parts, these ones you're seeing here are these. So these are called stems. These are called stems. So there are a total of 4 stems, as you can count with me. 1, 2, 3, 4. This is not a stem, this is a loop, just that I drew it as if it looks like a stem, but it's a loop as well. So we have 4 loops and 4 stems. You get? So why do we have loops? Why do we have stems? So these stems are actually regions where we have complementary bases. You get? So they go on to form base pairs and form the stems you see. Whereas this loop is formed because they are not complementary bases, so they repel each other. That's why we see these loops. So hope you've gotten the reason as to why we have loops and stems. Then we can go on to name these loops and stems. So from up here, we are seeing the 5' end and the 3' end. So this stem here is called the acceptor stem. So right, that's known as the acceptor stem. Acceptor. So that's the acceptor stem. Then this loop here is known as the D loop. It's called the D loop. So that's known as the D loop. And then this stem is also known as the D stem. Moving down here, we have we can see 3 lines that I tried to draw here. So this is known as the codon loop. So it's known as the anticodon loop, where which will go on to combine with the codons on the mRNA. Then this loop here, it's what we call the variable loop. So this is the variable loop, as you can see. This is known as the variable loop. And lastly, we have this loop. It's known as the T psi. I don't have the psi symbol, but you can have it in your you can have it noted. Let me just draw it. So it's known as the T psi, that symbol, C loop. We are going to look at each in depth. So T psi loop, and this will be the T psi stem. So what do these individual loops consist of? So these individual loops, what you need to know is that they contain unique bases. There are a variety of unique bases. However, we recognize mainly 5 main unique bases in tRNA, but there are more than 5. There are very many, as we are going to see. So in the acceptor stem, we have just the usual bases. On the 5' end, we have usually it's guanine, ending that loop. I mean stem, it's guanine. That's the nucleotide we have. Then, yes, the base. Then we have on the 3' end, we have from up, we have adenine. And then we have cytosine, cytosine. So it's CCA from the 5'. If you're coming from this side, CCA. If you're coming from the 3', it becomes ACC. You get the point? Yeah. So that's the acceptor stem. And looking at the acceptor stem further, we need to take note that it has usually 7 base pairs in this stem here. 7 base pairs. So it usually has 7 base pairs. So that's it about the acceptor stem. And what's the importance of this acceptor stem? The acceptor stem is where we shall have our amino acid added to this 3' end. It's where we have the amino acid added by an enzyme which we call the amino acyl tRNA synthetase. I can have amino acyl tRNA synthetase. So synthetase, you can look at my video about translation where I look at this in depth. So tRNA synthetase will come in and bring the amino acids to this stem. Also take note that there are about 20 tRNA synthetases because each is specific for a given amino acid. However, as I said, in some organisms, I can find that this tRNA synthetase is specific or is not specific to one, but can identify more than one. So you can have fewer synthetases. Moving on to the D loop. The D loop mainly contains a base which is known as dihydrouridine. Dihydrouridine, represented by letter D. That's it about the base. Then its importance is that it will be the recognition site for the synthetases. The tRNA synthetases will recognize the D loop. You get? That will be the recognition site for these synthetases. Then the anticodon loop will be the one which recognizes the base. So the codon on the mRNA. So if this is our 3' to 5', and it has, let's say A U G. So what will be the codon that will be read? The codon that will be read will be in the 5' to 3'. In other words, it will be in the parallel direction. So what will be recognized will be the U, A, C in the unparallel. This is 3 prime to 5. Then the codon will be in the 5 prime to 3 prime direction. That is for the codon. So also this uncodon loop has some unique bases, which are about 3. The first one is not denoted by letter Q, known as queer sign. Queer sign. Then it also has another base, which is known as inosine, denoted by I. Another base is what we call 7-methyl guanosine, written as M, 7 as a superscript, then G, 7-methyl guanosine. So these are the main 3 recognized bases, unique bases in this part. Briefly talking about inosine, the importance of this inosine, we shall later on see that it's very important for the Wabo base pairing, whereby it can pair with U, it can pair with A, that is adenine, it can also pair with C, the Wabo base pairing. So talking about the Wabo base pairing briefly, this is supposed to be under translation, not under structure. So this Wabo base pairing, you'll find that we have this base that can pair with more than one base in the codon. You get? So let's say we have inosine. They are normally found on the first in position 1 of the anticodon. In position 1 of the anticodon. That's why we normally have these unique bases that can cause the Wabo base pairing. You get Wabo base pairing, meaning it doesn't pair with one, but it can pair with more than one, which means that you find that one amino acid can be coded for by more than one codon. You get? That's the Wabo base pairing. It's position 1 of the anticodon, whereas it will be position 3 of the codon. How? Because if we have our anticodon like this, and this is our position, let's say this is position 1. When you look at the codons, we are adding from, let's say, from the 5 prime to 3 prime. So if we have used these 3, we find that the sequence that will be read next, or the codon, the set of codons, if we are considering the Wabo base pairing, it will be this base, the third base, it will start from here. You get? Because it is position 1 of the anticodon. So the position 1 will come here, and then plus these other 2. So it's the first codon, it's the first base of the anticodon, but the third base of the codon that are involved in the Wabo base pairing. For more information, check under translation. So, can look at the variable loop. The variable loop, mainly contains the unique bases, and not so much talked about, but we have what we call 5-methyl guanosine, and maybe the details you may need about this is that it has a length of about 3 to 21 nucleotides. However, it can also vary in the classes of tRNA. We have class 1, which contains short variable loops, and we have class 2 of tRNA, which contain long variable loops. So they are not so much of importance as that, except that you will need them when you're forming the tertiary structure. And then we can look at this T-Psi-C loop. So T-Psi stands for thymine, pseudouridine, and cytosine loop, or you find some books calling it the ribothymidine, pseudouridine, cysteine loop. You get? So that is it. But you can take thymine, pseudouridine, and cytosine loop. That's why you'll find some questions telling you the unique base that's found in tRNA. It's thymine, which is found in tRNA. So what is the importance of this tRNA loop? It's important for ribosome recognition. This is what is recognized by the ribosomes. That's the loop that's recognized. So with that said and done, we have covered the secondary structure of tRNA. And lastly, I will talk about the number of nucleotides in this tRNA. So it's about 70 to 95 nucleotides long. Normally, it's about 95, I mean 70 to 95 nucleotides long, and that's the secondary structure. So briefly talking about the tertiary structure. The tertiary structure was discovered by X-ray diffraction. Oh, sorry, crystallization. X-ray crystallization. Crystallization. You may not need the years and so on, but it depends on the examination body. Some will focus on the years and the people that found, but you can remember one person is called Rich. You can go and look for the other names, which I may not give you. They may not be so much important, but one person is Rich, and then the year was 1974. Okay, so that's about the tertiary structure. And what's the shape of the tertiary structure? We've said it's what? L-shaped. So how is this L going to be formed? The L shape is going to be formed in this way, in that we shall have, let me change the color here. So we shall have our tRNA loop interacting with the acceptor stem, and then we have the D loop interacting with the anticodon loop, forming an L-shaped something like this. This is just simple. Then we have the variable here. So this side we have the acceptor stem and the tRNA loop. Then down here we have the D, and it's not actually like this, but I'm just trying to demonstrate, and the anticodon loop on this other limb. So one limb has the acceptor and the tRNA, then one limb has the D and the anticodon loop, while the variable remains there in space. So that's the tertiary structure. So one thing you need to know, the forces that help stabilize this tertiary structure, which are very important. So the first one, you have H bonds. Those are hydrogen bonds. So where are these hydrogen bonds? So we looked at the pseudouridine that was in this tRNA loop. That's very important in the formation of these hydrogen bonds. Then the other one is what you call base stocking. Base stocking, or some books will call it the long base stocking, just an interaction between the bases in the different loops. So briefly, that's what you have to know about the structure of tRNA.